So I think John is going to talk to us somewhat anyway about how they got there, what actually happened in DeepMind in, I guess it's roughly 4 years from 2016, maybe 2017 until 2020. A really remarkable story, I think. So, John, over to you.
Great, thank you. It's really exciting. By the way, I guess, yeah, really excited. I'm sad, by the way, that I can't be there, but it was a little too tight actually to the Stockholm travel for me to feel safe going to the Dominican Republic. But yeah, when John asked me to talk, I was thinking, what should I talk about? And I realized we haven't really that well told the story of really, maybe not even AlphaFold1. AlphaFold is really, how did we get from AlphaFold1 to AlphaFold2? Great. Now, so the story I really want to talk about is the ideas in the middle. How did we, what was the succession of ideas? Because AlphaFold2, as I'm sure everyone in the audience has noticed, is a really, really complicated system. And it's not that we got there in an entirely straight line. And what I kind of want to talk about and discuss is what really happened, especially in this 2-year period that I'm more familiar with. And really the start of the story is at AlphaFold1. And AlphaFold1 was a wonderful system announced at CASP-16. And it was, I would say, a very good version of what a lot of the field had started to do in terms of deep learning in proteins. And it really was all about starting from the co-evolutionary information, in fact, the outputs of pseudo-likelihood, and running it through a computer vision network. And I really mean a network that we had reused the architecture from another computer vision network. And it was trained very well, and it was a very deep network. Apologies, just 1 second. Let me close this one. But it was ultimately kind of computer vision applied to protein structure. And what was really, I think, for us getting into it was really finding out, how do we use evolutionary information? How do we make these predictions? And kind of one of the big things that we had found that was useful is that instead of predicting contacts or just binary, are these two residues close, we started to predict distance. And we would predict distances. And originally we had this very, very complicated, and about half of CASP had this really, really, really complicated system for turning those distances into structures. It involved another generative model that was a VAE that was actually originally designed. This VAE was, maybe we could do an end-to-end folding system with VAEs, but it wasn't that good. So we would instead take fragments from it and use it in proposals for a fragment Monte Carlo of the type going on in the field and all sorts of complex things. And really, I think it was a system where we were trying to do this kind of simple deep learning in the middle, or at least straightforward deep learning, well engineered. And then around the outsides, we were trying to, do appropriate protein stuff. And we were trying to, think hard about the problems of search and optimization and everything else. And actually, maybe, and I had kind of come into this from more of a physics background and thinking, well, this doesn't feel very physical. It's not entirely satisfying. And I remember, looking at all this complexity and thinking, wow, why is this so complicated? Why are we spending so much time on the search of this, but not any search that looks like how proteins really fold? Not any kind of molecular dynamics. And I remember playing with some ideas in terms of Langevin dynamics on this and some others. And then it turns out, well, actually, all of that complexity we had outside the neural network was actually not worth much. And if we replaced it with repeated gradient descent, then we got really as good, actually slightly better answers from just plain old gradient descent and torsion space. And this was in some ways kind of weird. All these, thoughts and stories we had about search and Levinthal's paradox and exploration all kind of didn't really apply once we were holding these distance predictions from the neural network. And I think that, and you can see one, I think that was a video I actually showed at CASP of predicting the structure of some target. Now, what I think we drew from this, and kind of one of the stories I want to tell you in this talk, is really how did we kind of learn from what we saw in one system to build the next. And one of the things that I think a lot of people missed when you look at these distance prediction systems, and the system would predict about 64 by 64 residues at a time, and then we would assemble that. But for a small protein, that entirely fits within the window of these distance predictions. But I remember there was one kind of almost intellectual contradiction in how we thought about these, coevolution, convolutional systems and what they did in practice. And I remember this was a big discussion in the group where, I was saying, well, okay, if you think of these big, deep neural networks as denoising convolutions, which is how denoising coevolution, which is how people thought about it, then it would make sense that you could get a better understanding of which were true contacts and which weren't true contacts. But why were we predicting distances all the way out to 22 angstroms? Sure, there could be evolutionary information that two residues are in contact, and in contact can be a bit long ranged when you consider we're talking about C beta distances instead of, nearest atom distances. But more than that, the network was actually reasonably often quite confident about distances that are pretty far away. You can see on the left graph that we have distance predictions in blue and the true distances as red spikes. And you can see, for example, for the back distance, that it's sure those two residues aren't near to each other. They're not within 6 angstroms, but it's pretty sure they're at about 9-ish angstroms. And in fact, I think we saw even some examples, I remember, where it was pretty sure the residues were at 13 angstroms. And this was really a big mystery. How can, if this is really, understanding or denoising evolution, how can it be sure that two things are in 13 angstroms away? What could be the evolutionary signal for 13 angstromness? And the only real way, it's kind of like being sure that your friend is 180 meters from you. You're only sure your friend is 180 meters from you if you really know where they are, and then you're just expressing it in a distance because that's the output. And it kind of became clear that in very favorable cases, maybe this network really did find the structure in the middle of the network, but then of course it had to produce distance outputs. So it would convert that structure back into distances. You had this chain of geometric information that would make it really, really certain that these two residues were 13 angstroms away. And so it kind of, we were, as we started to analyze this and as we were looking at the results from CASP-16 and looking at the predictions in general, it kind of became clear that sometimes the network was really solving the structure. And that was part of the reason in which we were able to do something with gradient descent, is in fact, all of this great geometric information had been discovered by the network. And I think it was one of the first clues to us, and I'll say throughout this talk, by the way, try and remember and say what we were thinking. And, you could probably find references or something else that says we should have known this, and that might be true, but I'll try and say what we were thinking. But there was this kind of sign coming out of CASP-13 that these neural networks are doing something more than denoising evolution, that they really are understanding the geometry in a relatively deep way, and we were probably not exploiting that very well. And there was a lot, it worked on 64 residue fragments. There was a lot that was probably wrong with how we were handling these distograms. But there should be something really great under there. And kind of coming out of CASP as well, I would say we were all, and this actually, I should say, coming out of CASP, as you all know, there's the 3 months of doing CASP, and there's the 3 months of waiting for CASP afterwards, right, to find out the final results. So this was in, mid 2018 or somewhere toward the end of CASP. Dates are rough here, but, came out saying it didn't really feel like we were progressing that far. AlphaFold1 was a great system, but we kind of felt like we weren't just going to extend it and extend it and extend it and get out to a really great prediction system. And I remember, we decided, and I was kind of coming, I said, we've got to learn physics, right? We've got to do something with physics. And we decided, like, one of the ways we were going to do that is we were just going to stop doing evolutionary structure predictions. So kind of as CASP was wrapping up and we were making these predictions internally from a research point of view, we said, well, what happens if we just don't let ourselves use evolutionary information? And it had that also nice side effect that we had put a ton of work into this CASP-16 system, and how do we, really, really tune this? And, it's this giant, like 600 layer ConvNet, if I remember correctly. And that was really a pain to work with. It was so much of a pain that people only tried small ideas on it and wanted to get to a situation. Also, handling MSAs, as everyone in this audience knows, is a pain. And so we said, how are we going to get away from this and really do physics? So we just said, okay, well, we're gonna do the same kind of leaderboard of pre-modeling accuracy, but we're not going to allow ourselves to use evolutionary information at all. And started to explore different meanings of what that could be. And there were different approaches. A lot of them were more search-based, or some more MD-based or learned MD type approaches. And I had been excited about that for a while. But there was this other thing that had kind of, gnawed at least me, is there was no geometry or there was not a very physical basis of how we were handling proteins. There was really not that much proteiny in our big histograms. In fact, the geometric information was spread all over the histogram, right? So, each point in there represents a pair of residues. And if 3 residues come together, the information about the distances in those 3 residues could be spread all over this histogram. Could be dozens and dozens of layers before they can have any influence on each other. So all of this kind of felt wrong. And attention was, pretty clearly becoming popular in general. And like during this period, which was really kind of in August, started to play with these ideas of, well, what if we build a geometric attention? And kind of called it covariant attention at the time. Later, it became invariant point attention, which is a much more accurate name. But the kind of first place that we were doing this, I was playing with actually sidechain prediction, because I had done this task in my thesis, and it was kind of an interesting toy task to get an intuition for how learning works. So it's kind of fixed backbone sidechain prediction, squirrel-like. And this worked surprisingly well. Like almost immediately, we could predict sidechains really, really well, and much, much better than anything I was doing in my thesis, and just kind of comically easily by setting up this geometric attention and then asking it for the sidechains, and it just worked. And then it also forced the development of this frame idea, this notion of local geometry that was the core of the structure module. And relatively soon after this, within maybe a month of having geometric attention, started to think about, well, could we really turn this into maybe an actual folding system? Instead of worrying at all about, how proteins really move or how it should happen, could we just get the network to update positions? And in the bottom right, I don't know if you can see my pointer, but that kind of weird image that you see of embedding, right, analyzing and folding was how we were originally thinking about it. It was this notion of geometrization. It was kind of this idea of, well, maybe we can really think about the ensemble of proteins, and then we can shoot off a couple realizations, and then we can analyze those realizations and embed them again and do this process iteratively. And started to think that maybe that's a good idea. And actually turned out to be closer. I didn't think about it until preparing for this talk, but it actually ended up kind of close to what AlphaFold looks like when you now do recycling. But started to think, well, maybe this would work. And the other, kind of important insights, and I left one off, is, well, maybe we don't need to think about torsions. I remember the other thing that we're thinking a lot about is, Well, we were doing all this work trying to stay on the physical manifold of things that look like a protein, right? And keeping all the bond distances correct and keeping the angles correct. And that basically forces you to work with torsions, and torsions are awful because of lever effects. What if we just didn't have those? And so started to say, well, what if we just said all of these are rotational frames? That's kind of what we would call the residue gas. And we work without that. And so we had this frame representation. We were thinking about iterative updates. The one, and I put it in bold, is after a little bit of playing with LDDT and it not working very well and getting lots of weird chirality things, I said, well, what's the simplest change we can make to LDDT, to a pairwise distance loss on structures that respects chirality? And almost immediately FAPE came to mind, which we had called affine LDDT, which it's not really affine, but it's kind of rotational LDDT. And when we were doing all this evolutionary free work, this kind of basically the structure module was almost all at once comes out of this. And to be clear, the results were absolutely terrible. They were like 30 GDT systems, but they weren't 20 GDT systems. And when you looked at the structures, they looked like proteins. They looked like reasonable wrong answers. They had helices, they had sheets. And it kind of became clear that this seemed like a good idea. And after maybe a few weeks, a month of working with what was basically a structure module on the evolution free problem, Rich decides, well, what if we just add the distogram in as another input, as another 2D input? And almost immediately, as soon as he does that, and we use the CASP-16 distograms, we see really good structures. I mean, almost one, maybe 2 GDT worse than what we had in CASP-13. So pretty immediately, in a system that is basically run CASP-13 distogram network and jam that into what we would later call the structure module and run the iterative refinement, use FAPE loss, by about October. And so I remember this distinctly because this was before the CASP conference. And I had kind of jokingly said we should try and research to have one amazing thing that we can't talk about at CASP-13. And that turned out to be direct structure prediction. And it was ever so slightly worse than AlphaFold1, but it was still, we thought, a really, really clean sign of, wait a minute, we can just go all the way out to structure. We can skip all of the Monte Carlo. We can skip all of the search. And we didn't train it via end-to-end gradient descent, but it looked really, really likely that we knew how to go straight to structure. And I would say, though it wasn't more accurate, it meant that all our experiments now became really, really fast and cheap. And we started working more and more in terms of, okay, well, we can try this. We can get answers immediately. We can start to iterate and very quickly prove and disprove ideas. Of course, we go to CASP, really interesting. In the meantime, we're trying really, really hard to improve this structure module system, right? And since the distogram was really painful and we were still using the AlphaFold1 distograms, in fact, just saving them, not even rerunning the network. And we kept trying all these ideas to make the structure module better. And what we found is that every single thing we tried did almost nothing. So we got into the first, and even across AlphaFold, there were a couple of long periods of kind of stagnation, I would say. And this was one of them, where everything we would try would not really make the results better. They stayed 1 or 2 GDT worse than the AlphaFold1. And we started to refer to it as the distogram barrier, that it became pretty clear that most of the really good stuff was still happening in the distogram. The structure module was realizing a good version of it, but the structure module didn't have the ability to kind of transformatively improve it. We did ultimately get a little bit better than AlphaFold1 when we put sidechain prediction, the first versions of sidechain prediction, Russ and Alex had done that, into the structure module, but it was still a half GDT better, something really, really small. We weren't really doing something really, really good. And so from here, we started to say, well, what are we going to do next? How are we going to go beyond this? And it became really clear, well, if we kind of tried to, if we rethought how we build structures and got rid of the gradient descent, well, now it's time to think again about how do we use evolutionary information. And this was really late 2018, early 2019, maybe January or February. BERT and mass language modeling was very popular at the time. And we started with, doing single sequence protein language models. And in fact, Karen on the team had done this even before AlphaFold1, but the results were disappointing. And I think in hindsight, they were much too small. It was before scaling was well understood. I can't remember how many parameters they were, and they were undertrained. But also, it kind of didn't quite feel right for this, is that we know that the MSA is really useful information. We were really thinking about how do we use this and how do we not do the really annoying kind of pseudo likelihood and coevolutionary calculations. We've seen, I think, Bjorn's raw MSA paper, although I'm not sure how much we thought about it at the time, other than convolutions were weird to use in it. But started to play with, and I think this is one I was playing with, of, well, what if, there's a really clear connection between pseudo-likelihood-like objectives, what is the residue given a linear model on all the other residues, and BERT, which is saying a non-linear, you can think of mass language modeling on MSAs as being basically non-linear kind of pseudo-likelihood. And so we started to play with, as a totally unsupervised objective, and the first one was, how do we do mass language modeling on top of the MSA? And so we would call this raw BERT, right? Raw MSA BERT. And the other thing was that wanted kind of semantic operations. And on an MSA, of course, the rows are very semantic. Those are the sequences. The columns are very semantic. That's the same residue across evolutionary trees. So we just did essentially axial attention. We would attend across rows, we'd attend across columns, we'd tell ourselves a very semantic story about what it was, but that was what we ended up using, and it worked really, really well. And it started to have quite reasonable MSA prediction results. And we said, okay, let's just hook that up to the simplest thing we can do. And we stuck something like a 10, 20 block convolutional network on top with a distogram objective. I can't remember if we had a stop gradient or not, but we would basically do kind of an outer product, and then we would do a convnet. And that almost immediately produced something better than our AlphaFold1 distograms. And so this started to look really, really good. We were doing just straight distogram prediction here. And so it became, I think this was this moment at which it started to become really clear that we could throw away kind of a lot of the complexity. We can go back to raw MSA. The convnets were still in there, but this really started to work. And we started to really, think that using raw MSAs, using this MSA objective, which really turned out to stabilize the training of the distogram and the convnet, that these things would tend to fall apart if you didn't have the MSA objective. We've since been able to get rid of it. But that started to really be the moment at which the second ingredient came in, is we know a different way to handle evolutionary data. And so the obvious next thing to do, and the first thing we did is, well, okay, let's, this is much cheaper, and we no longer have that AlphaFold1 thing that we didn't really want to train anymore. So let's just hook it all together and literally hook it up into the structure module. And instead of using the distogram as an input to the structure module, we'll just take the pair activations from this network. And so we would run this axial attention on the MSA. We would run, and it would have a loss. We would run the convolutional network, and it would have a distogram loss. And then we'd go run the structure module. And this really worked beautifully. It was kind of the first system that looked a lot like AlphaFold as it would be. And essentially from here, it was also super, super fast to train, easy to work with. Everything about it was kind of painless, which meant it was really, really easy for everyone to try their own ideas on top and to stack on top of it. And when you're working on a team, and we're working on a team, and we're thinking about, a team objective, you need to stack on each other's ideas. And we would just basically, everyone would be building ideas into this. One of the more consequential ones, and this actually happened, we saw the sparse transformers paper come out that said, well, if you're processing images, axial attention or crisscross attention is a good idea. And I remember thinking, wait a minute, we're already doing axial attention, but we're only doing it on the MSA. And if you think a little bit about the pair matrix, axial attention sounds like a good idea there too. Instead of having lots and lots of convolutions on every step, what if we just took the same module, literally, called it a second place and replaced the ConvNet with axial attention? And that worked dramatically. Suddenly the network was learning much, much faster, was learning really, really interesting things. Actually, at some points we had alternation of convolutions and axial attention, and we found deleting the convolutional blocks without even adding more of anything, of any other type of attention. Just deleting the convolutions made our accuracy better. And it started to become clear that convolutions were actually the opposite of what you wanted for structure prediction, at least in this setting, or at least that was kind of our intuition, because they encourage the network to think locally about primary sequence. Discourages thinking about kind of larger tertiary interactions, distant contacts that are just as accessible in axial attention as anything else. And so this started to become one of these intuitions we had, is that really you want to be careful actually how much you force the network to think locally or think about something other than the tertiary interactions and the physics. And I think it makes sense now that you think about it, if you think about kind of a graph interpretation, if you think about wanting to have kind of every layer doing meaningful information propagation across arbitrarily large sequence distance. But it was quite the surprise at the time. You never in deep learning delete a layer and then simply get more accurate. The other kind of major idea we had at the time, or what we felt was kind of uncomfortable about this, is that we were thinking a lot about, well, we're doing MSA processing and then we're kind of throwing it over the wall via an outer product. And then we're doing kind of what we would think of as the physical processing, ignoring or having the evolution only kind of carry that information forward. Then we do the physical processing for a while. And then we do the geometric processing of the structure module. And it felt like maybe we should, put these together. And it was actually Michael Fagrinov who had the really good idea of, well, what if we just, instead of running this as phase 1, phase 2, what if we just run them as, we would call them 2 towers. I guess we always write it sideways now, so they don't look like towers, but 2 tracks if you want. And this was the birth of the first thing that really looks like an evoformer. Instead of having all the kind of sequence or evolutionary processing followed by all of the physical or pairwise processing, we'll just run them together and we'll run a sequence of layers. And this originally was only worth a little bit, but as we built ideas on top of it, it became worth more and more. And maybe more importantly, we thought less about, say, the ratio of how much MSA operation to how much physical operation. We just linked them together. And so this really started to be the point at which they had kind of conversations, and it looked, more like, okay, well, we invented operations to link them together. We had the pair biased attention, which we basically took from the structure module and brought that all the way back to the MSA processing. And then the outer products we just had a lot more of them. And that really made it quite a bit cleaner, and we could understand and appreciate what it was doing, and we could see the evolution of this information. We also at this time, and this is a story maybe a little bit out of historical order, because Richard started to play with this when we were already in the kind of Evo-free mode, but self-distillation came across. And this absolutely felt like magic, like cheating, that we'll predict structures, we'll take the confident ones, and then we'll add them to our training set. And later we found out about the noisy student paper, but it was a bit of an independent coincidence. And it arose, I think, maybe first as cross-distillation, but then it started to become a quite important part of AlphaFold training. And it still is a relatively interesting thing to think about. Where does the information, it's more accurate. You would presume there is extra information available. It's not absolutely clear where that extra information comes from. You can say, well, you don't augment or you don't add as much noise as you do when predicting to make training data as you do as you predict sequence subsets of proteins or others. But I think it's also about the time to the first time you repeated data. Of course, we have so little PDB data that we need to treat it really preciously. And I think it's probably more about, the early training dynamics and getting it to really think about tertiary structure. But this was really a surprise, and it kind of popped nicely on top of our networks. And almost immediately we said, well, we'll iterate this multiple times. We'll keep getting better and better. And we found out that was mostly not the case, that in fact, the quality, a couple extra GDT in the network you used to make the predictions didn't have that much impact downstream. So that's why we ended up with a fairly old distillation set in the paper. Now, this other great insight, and this was actually Alex Britzel had, is, well, if you think about the pairwise interactions as kind of graph-like, or you think about, well, ij edges, right, in a row attention, or attending ik edges, right? Ij is talking to ik. Well, that's kind of two pieces of a triangle, but there's a third piece. Surely this jk interaction was going to be relevant. And I remember kind of Alex telling me and saying, this should be a good idea. And then it became really clear that there was a very, very simple way to implement this idea. Well, we'll just take our attention and we'll just pair bias it again. Pair biasing worked great for the structure module. It worked great for the attention on the sequence. Let's pair bias the attention on the pair matrix itself. And there's another natural way to think about it, and kind of a quantum problem that comes up is what's called in-representability. If you have a pair matrix, could this actually, if you think of the distograms, are those distograms consistent? Could they represent the real distograms of a real set of beliefs about what the structure might be? And the answer is often no. And we went back in response to some reviewer question and said, when you train AlphaFold, how often does it violate things like the triangle inequality? And the answer was that not a huge fraction, but some reasonable fraction of time, it would violate the triangle inequality, that your distogram beliefs were inconsistent. And adding these small kind of triangles, adding this pair biasing, actually reduced that by about a factor of 4. It really did give the network more, I think, 3D consistency. I don't think you should think of it as only implementing the triangle inequality. It was kind of, we did think about the triangle inequality, but then we generalized it. We said, okay, well, let's just put in the triangular interactions. Now, after we did this, and of course, as you all know, AlphaFold's still pretty large and painful and expensive to use. So we were trying to figure out a way also to make it cheaper. And Alex had the idea of, well, what if we just get rid of the attention and do these, I think what we ended up calling multiplicative updates, but these kind of three-way graph interactions with no attention. And it was not quite as good. It was very, very close, but not quite as good. And we did actually use this in the middle to build a no attention AlphaFold, basically removing all attention, and it still worked about as well. We noticed these interactions weren't quite as good, but then at some point, Alex tried adding these as well and found that alternating the attention and these multiplicative updates was slightly better than doing attention only, or even doing twice the attention. And so if you see this kind of weird thing that we have attention and then multiplicative updates in the evoformer, it's because we found out both were better than either. And it was a pretty empirical fact. So evoformer ends up having both. And so this was a lot of the ideas, I would say, and a lot of the iteration that got us toward AlphaFold. But recycling was actually one of the last to come in. And this one is pretty directly due to CASP. And more specifically, that CASP asked for 5 structures. And of course, our network was only producing 1, and sometimes it was quite low diversity. And so the idea was, well, maybe what if we just run our network and then we take the output structure of that network and we make that an additional input, and then we'll give it a loss that's maybe the best of the 2 structures, and that will encourage it to make diversity. In fact, we had some other attempts to make diversity by running 5 structure modules. Now, before we did that, I was sitting there going, well, we probably should run a simple experiment. Let's just put the structure in and just ask for the same output and see what happens. And that turned out to be a reasonably large boost in prediction accuracy. So recycling was basically the baseline for this diversity experiment. And we ended up, I think, never really finishing the diversity experiment because recycling became pretty clearly almost as good as adding layers. And this is kind of a weird thing if you think about networks in terms of memorization. If you think about networks as running gradient descent, we would later produce these plots that would show GDT over layers that would start to convince us what was really happening here was that AlphaFold was doing a lot of computation. It needed layers to give it compute. But that was kind of how recycling came about. It was a very kind of simple thing in terms of giving us synthetic depth. And we started training, 4 block AlphaFolds with, 10 recyclings and everything else. We settled on 3 recyclings for CASP, but it was pretty clear that it was almost but not quite as good as depth and a lot more convenient to use. And that's how recycling ended up in. And then Michael actually figured out, well, let's not just recycle the structure, let's also recycle the embeddings. And that worked even better, certainly trained faster. Kind of another part of the story is confidence. And we were always slow on both confidence and physical realizability of the structures, because it was always I was always saying, let's just solve this at the end, right? We can always handle this at the end. And we solved confidence once every time we needed to. The first time was actually distillation. We needed some way to throw out the bad predictions. And so we did this thing that we called KL confidence, where we would see how much the histogram differed from our reference histogram, what it would look like if the network had no idea or it was sequence independent histogram. And we had those sitting around, so we used those, and it had no real connection to physical accuracy, though you could calibrate it. But it worked quite well at filtering distillation. So that's why in the AlphaFold2 paper, there's this weird KL confidence for distillation. I think some people assumed it was a great strategic masterstroke, but it wasn't that. It was just that was what we had made at the time. And the kind of second time that we went after and did confidence was talking about PLDDT. And this arose because we wanted to make predictions of SARS-CoV-2 structures in the early days of the pandemic. And we really, we were pretty sure we had not quite the finished AlphaFold2. I think we had almost everything but recycling at that point. But we really wanted to make sure we were releasing structures that would actually help. And the idea came pretty quickly of, well, LDDT is a good measure of quality. What if we just regress that? And we just did the dumbest possible thing where we put a single head on top of AlphaFold, fine-tuned it, and found it really quickly to produce accurate LDDT. So the predicted LDDT became PLDDT just in time for CASP. I believe we used it as a CASP ranking metric as well. And then the final one, PAE, was actually not present till much later. We wrote our paper, we submitted the first version, a bunch of really great reviewer comments came back. And one of the comments was, it's all well and good that you can predict LDDT, but that's a local measure. It can be deceptive. I like TM score. Can you predict TM score? And, we were about to write no, but before we wrote no, we decided to sit down and think about it. And at some point kind of realized that, well, what you need to predict TM score, if you think of this as an aligned score, it's about to say no because you can't align. But of course, we have kind of residue-like alignments. We're already doing that in FAPE. And FAPE was one of the really, really important parts of making this network work. So if we just kind of predicted the components of FAPE, what we started to call the absolute error, and so then PAE is PAE. Then we could actually predict TM, we could predict GDT, RMSD. I mean, you could still do that. We derived them in the SI. And probably more importantly is this finally gave us a way to talk about pairwise error. And I remember even talking to CASP and saying, well, it doesn't make any sense to ask how accurate is a residue. It's not true in isolation. It's really, an LDDT is a bit of an environment score. But what really makes sense is to say 2 residues are right relative to each other. And this actually fixed another problem we were having in the AlphaFold database. We were making it, we were talking to Dimble, we were saying, oh, well, we need to put in all these histograms so people can interpret whether the domain-domain interactions are correct. And when we would try it with scientists, they really couldn't read histograms. And they're honestly kind of hard to read because a single contact really changes your opinion of what's a confident interaction. And then suddenly we put in PAE and everyone could read it immediately. And so it became, I think, the right way to do it. And it really all just came out of a reviewer comment. So thank you. There are even more ideas, and I really don't have time, and I'm probably over time, but to describe all the other ideas. Early in AlphaFold, for example, any structure longer than we had at training would immediately collapse. And then we figured out how to do this relative positional encoding. We figured out how to put in extra sequences. How you initialize really matters. Metagenomic data, including BFD from Martin, right? How do you templates, gated updates? How do you solve violations with violation losses primarily and gradient descent? And there are many, many more. And all of these are stacked together in part because we had this kind of stable base of a very, very fast, easy to use system that it was very easy to measure these small changes. And maybe the largest single change was distillation at something like 2.5 GDT. Everything was a really, really small change that ultimately added up. I'll also say CASP, in addition to all the great things it does in measuring how good the field is, really does make your systems better. I remember we had two targets that we learned a lot from. One was ORF8, which was terrifying because it has horrible genetics. It's an incredibly fast mutating protein. It has, something like 22 sequences using our MSA tools. And we fixed it in a weird way, and we ended up using this multi-head system and something else. But what we hadn't realized is that if we had just recycled more, it probably would have fixed it. I think the more interesting case was really this giant polymerase. I feel like it was H1044 that CASP had released in each of its parts, and something like 25% of CASP was this RNA polymerase. And it was about 2,000 residues. And AlphaFold couldn't really handle it at all. It would give really, really bad predictions. And the first version we had was we'd predict the individual domains and then we would patch them horribly into the templates. But then we started to realize, well, the problem was that we trained at 256 residues and it was just a bit too big. And so we retrained AlphaFold or fine-tuned AlphaFold at 384, and suddenly this worked really, really well. And we started to understand that AlphaFold2 would go at about 8-ish times the training length before it started to collapse. And so the model we ended up releasing was the 384, and it was a much more useful model, really thanks to this particular target. And we took that lesson into, AlphaFold, especially AlphaFold Multimer 2.3. We also did a really inadvised ensembling over CASP that we measured later as being worth basically nothing and just made it 8 times more expensive. So we definitely made mistakes. But CASP forced us to think really, really hard about each target. And we were doing almost exactly the opposite when we were developing AlphaFold, is we didn't want to look at each target too much because we were afraid we would overfit to those particular targets. And I think it was a really interesting exercise. Both AlphaFold1 and AlphaFold2, the course of doing CASP taught us how to improve our systems. Now, which part of this matters? And I want to say, really, as a scientific community overall and as an ML community, we tell too narrow stories. We like our hero narratives of one particular thing. And you go look at the ablations, you also look at the progress of AlphaFold2 over CASP, and these stories are too narrow. Everything matters a bit. Sometimes you solve a problem multiple ways. Often what we think are really, really important turn out to be less important later. Lots of people got really, really excited about AlphaFold3 not using equivariance in its equivalent of the structure module. And I was always mystified by that, because if you look in this table that everyone saw in our AlphaFold2 paper, we had this row that said no IPA, use direct projection. So all of the geometric operations that, all right, we're very excited about was kind of there at the kind of first recognizable component of AlphaFold2. But we had already shown those were worth a couple GDT to AlphaFold2, that if we didn't have those, no one would really notice. I think we get so excited about the heroism of let's put geometry into it in exactly this way. I think we put geometry into AlphaFold in 5 or 6 ways. And the IPA version of it was the least important of those. And I think we need to think more. And as we also understood from AlphaFold1, we really understood there was some amount of direct structure prediction going on. There are a lot of lessons in the systems if you listen to them really carefully, if you really take an open-minded approach to the data. And I think we can do that. Similarly, kind of the change we had in the amount of how the evoformer worked versus the paraformer was really inspired by the covariance somewhere in here, the no raw MSA ablation, which was really quite good. And one of those was really, really good. So we started to see these lessons, and there's, I think, a lot more lessons we can learn from these systems and make them better. And I'm running probably overtime, so I'll go quickly. I will also say that it's really, really important to think about, should biologists use our systems? And GDT is a really incredible metric for measuring progress. It's a genuine advance over RMSD or other things that we had been using before, and things like TM score about equivalent. But it doesn't tell you if the prediction is relevant. And the first time we did this, we were kind of in the development of AlphaFold2. It was maybe 9 months before CASP. We said, well, we're doing really, really well. We're at something like 85 GDT. I'm sure biologists will gain a lot from using this system. And so we decided to test that by looking in papers, by making structure predictions for these papers. These were announced papers with a new structure in Cell Nature Science, I think is what we had picked. And then we would look at the discussion section of the paper and they'd say, oh, we noticed the pore narrowed, or we noticed the tryptophan covered the active site, or some statement the authors made that they learned from their structure. And we would look at our predictions and say, would you have learned the same thing? And the first time we did that, the answer was really no. That an 85 GDT system typically did not, maybe 3/4 of the time, contain what scientists said they were really exciting. They would always look really quite reasonable in cartoon representation, at least non-overlaid, but they would be missing that essential aspect of the biology. And we realized that we really had quite a few ideas, and some of the ideas, maybe we had evoformer, maybe not, but certainly some of the triangular tension and others all came after this period where we realized we had to be much more accurate. I think it's something that we need to make sure that we understand whether we're measuring progress, and CASP does an exceptional job at carefully measuring progress and carefully comparing and deciding which methods are the most accurate in a really, really statistically valid and epistemologically valid way. But then the question is it good enough to use for conclusions on these tasks? I think we need experiments more like this. We need more expert judgments in narrow areas that won't necessarily come during CASP, because you need a somewhat longer time period to accumulate enough evidence for a particular task. And the second time we did this, it was much, much better. And it really also emphasized that those last few points of GDT mattered way, way more than you think. And you should really think not so much about GDT as 100 minus GDT. How close to experiment are you? The other thing we learned, and this was during the papers, we had some idea, or we thought even during the development of CASP, that it was maintaining kind of an ensemble of hypotheses. But we wanted to understand a little bit more of the network, and we trained the structure module per layer so we could project out these movies of how it really went. And we were shocked to see how kind of smooth and dynamical they were. And some of this may be an artifact of the residual connections, but I think a lot of it is really showing you that most of the time and capacity in AlphaFold is really spent on refinement. And that was the reason that there was a null result in CASP-16 for MD refinement of AlphaFold, which was somewhat surprising at the time. That was our first real hint that we were doing really well in CASP, because we kept getting sent out as refinement targets. But I think it comes down to, because the refinement is actually in the network and in the evoformer, not the structure module, that the evoformer is a very clear kind of geometric notion that it is using to do this refinement. And we started to understand that as we made these per layer predictions, and they looked so dynamical, and you could see different domains being resolved at different times. And I think there's a lot more to be done in understanding exactly how you move from frame to frame of this video and what other information may be there. But we can also dive into these, and I think we should be doing more of this and more prediction of intermediate layers, and start to understand, how do these predictions arise, when do they arise, and find actionable insights. This was one of the insights that led us to go to the performers, saying, well, let's spend more capacity on the geometric part of the network. I want to thank both the people, the two authors, CASP enormously, really defining this field, John, Andre, Christoph, the entire community, as, defining what we should be doing. And I think it's a real testament to this community as well, that, when the CASP-14 results came out and everyone in the community said, this is such a big leap and it's going to be really, really important, a lot of experimentalists were skeptical. They said, oh, CASP structures are easier, which we all know they're not, or well, it's not going to be, really relevant. And then 6 months later, when the when the paper comes out and everyone finds out, no, CASP was exactly correct. They exactly, said this would matter, and it matters. I think it's a real testament and foresight of the community and of measuring well. And I think so much biology progress is held back by not measuring well, by not understanding which ideas really work versus which ones we really hope will work. And the last thing, and I want to make a big emphasis, is the protein data bank. That we were able to do this work because more than 50 years ago, people had the insights to say that collecting the structures together is important, that data curation matters, that journals should, demand deposition to do this. And I think that kind of foresight 50 years ago is an absolutely exceptional part of being able to do this. We are all here now working in machine learning using data, and without those data and without the careful curation of it, we couldn't do this. Thank you. I'm done, but I can't hear anyone.
Thank you.
So hopefully you've all heard that presentation.
We're really behind, but I'll take one or two questions. Are we ready with the mics? Who's got a question? Question? No questions? I've got 20 questions.
Okay.
Hi, John. Thank you so much for the great presentation and the great contribution to the field. I have a question, not necessarily related to structure prediction, but we have seen recently many research groups using AlphaFold2 and 3 to predict the protein-protein interaction. What's your reflection on that?
I mean, there's two versions. One is really, really clear, where we use it to predict the structure of interacting proteins. But I think you're referring to interaction discovery.
Yeah.
I think of it as off-label structure prediction use. Is that really what these models are trying to say is, I'm this confident that if there's a complex, it looks like this. And then we're converting that into, do these interactions exist? I think what people are seeing is, it often works really well. I mean, I think, the ability to do it at scale, like the really nice cell paper with the fusion complex of egg and sperm, or fertilization complex. It often works really well. It's leading to insights that people confirm. I think, though, as a community, we need to think harder about the false positives. There's some nice work out of the Walter lab on Spock. There's some others. I think probably Arna, I think, has some work. I think we as a community need to figure out what to do about the false positives, because the network is trying to tell you if they bind this way. So it's entirely natural for homologs, if they bind, will bind the same way. And so we need to figure out how we're going to drive those false positives really low, because then we can start to get these huge interactomes that we all want. But I think we're kind of at the early stages of this. We really need to think more as a community about how to measure well, how to measure the same thing. Maybe it's something for CASP-17 to go into once we figure out what experimental truth is, which is unfortunately really slippery in this field at scale.
All right, I think we... I've got one more at the back.
Yeah, one more in the back. Sorry. Really nice talk. I had a question as an experimentalist, what your thoughts are regarding conformers that have geometric strain. So these are the 1 in 3,000 non-common rotamers, the 1 in 10,000 non-common Ramachandran outliers that play an important functional role in active sites and regulatory regions.
I don't know of studies at scale. I know of a bit of anecdata, and I'm trying to remember author of Isolde, and I've forgotten his name at the moment. Actually, I remember emailing us shortly after AlphaFold2 came out and said that we do at least sometimes predict correctly cis non-proline residues. And so we do predict some really strange stuff. If the rest of because remember that strain is also going to be distributed. The energy cost of that strain is going to be distributed in the rest of the structure that's going to be trying to force it into that conformation. And since AlphaFold is only somewhat physical, and a lot of it is like, what is everything else, trying to make it make sense, it should do reasonably well. It would be interesting, though, people should, I think, check more. It would be very interesting to just say for all the new, say, cis non-prolines or some of these other really strange geometries, what percentage does AlphaFold do well? My guess is a lot on backbone, maybe rotamers, maybe sidechain rotamers would be harder. Or at least, chi outliers might be harder, but I'm not sure.
Okay, I would like to go on, but really we've got to stop because there's more program. John, for me, this was really amazing. I've got a lot, as I say, a lot of questions, but maybe some other time. And I think everybody here was absolutely enthralled. So thank you very much again.
Thank you. Bye.